we can start. We are going to look at the third lecture series on our digestive system lecture series. And in this third lecture, we are going to focus on the extrinsic glands or organs of the digestive system. So in particular, we are going to talk about the salivary glands, the liver, biliary tree, and the pancreas. Remember where we've come from? The first lecture, we just looked at general concepts and anatomy of the upper GIT. We looked at general concepts of the digestive system and anatomy of the upper GIT, mainly focusing on the oral cavity, pharynx, esophagus, and the stomach. We didn't look at duodenum, although it's part of upper GIT. Second part, we looked at anatomy of the small intestine and anatomy of the large intestine. And that's where we looked at the duodenum as well. So in this part, today, we are going to focus on extrinsic organs of the digestive system. The other two lectures remaining will be largely on embryology and malformations of the digestive system. So in today's lecture, first, maybe we review how we classify the anatomical components of the digestive system, just in a word or two. Then we'll focus on the subject matter. We'll start with the salivary glands. Then we'll focus on the liver. We'll then look at the biliary system, and we'll finish with the pancreas. So let's back to our first agenda, just reviewing the classification of the anatomical components of the digestive system. As we mentioned, we can classify the components of the digestive system in two ways. We have the hollow GIT. When we talk of the hollow GIT, we are referring to the part that is usually followed by food. We call that part the alimentary canal or the gut. Remember, the gut will include on its wall some glands. The glands which are part of the hollow GIT are termed intrinsic glands. And those ones are not our target today. Those ones, we've already discussed them when you're looking at the first two lectures. So intrinsic glands are glands which are incorporated within the wall of the digestive system. The ones in the stomach are called gastric glands. The ones in the intestine are called the crypts of Lieberkühn, in the mucosa rather. And the ones in the submucosa of the duodenum are the Brunner's glands. So this just to mention a few of the intrinsic glands within the digestive system, as opposed to what we want to cover today, which are the extrinsic glands or organs. These are basically organs which are not within the wall of the digestive system, but they produce their secretions which enter the alimentary canal. They're not within the wall of the gut tube, but their secretions enter the lumen of the alimentary canal. Those are our subject matter. Now we can start with the salivary glands, and we aim at looking at the names of the salivary glands and also to state the location of each salivary gland. If you're able to know the nerve supply, it will be good, and the role of each salivary gland. Remember, salivary glands are named so because they produce saliva. The saliva is usually released into the oral cavity. There are two categories of salivary glands. The ones which fit today's class are the major salivary glands. The major salivary glands are the ones which are extrinsic, to mean they're not incorporated within the wall of the mouth. They exist in pairs. There are three: submandibular gland, sublingual gland, and the parotid salivary gland. So they're in pairs, which means you have right and left. Those are the major salivary glands. But saliva is also produced by the minor salivary glands. Minor salivary glands are intrinsic glands, which means they're within the wall of the GIT, and in this case, within the wall of the mouth. So which structures in the mouth really house the minor salivary glands? There are those which are in the palate, we call them palatine glands. There are those which are on the cheeks, we call them buccal glands. And there are those ones which are within the tongue, we call them lingual glands. Those ones within the tongue are also called the glands of von Ebner. The glands of von Ebner. Now, in general, whether from major or from minor salivary glands, we produce about 1.5 liters of saliva per day. Of course, that would vary depending on a number of circumstances, but that is in general. Saliva contains a lot of things, but we can look at the component of saliva as this way. There's the big part that's water, and when we look at the solids, which is really our interest, well, let's call them solutes, because now gases are not solids, but those substances which are within that water. So the gases, fine. You also have some minerals and quite a number. But again, we have some organic substances. The organic substances that we have, some are enzymes, which help in digestion, or enzymes which fight infections like lysozyme. But we also have other substances which are not necessarily enzymes, but they're organic substances. So in a nutshell, the saliva contains many things. If you look at that list, glucose is not in that list. We don't expect glucose to be in saliva, but if somebody is diabetic, they have high levels of glucose in the circulation, then we'll expect salivary glands to also excrete glucose. What are the functions of saliva? Having mentioned that there are many things there, I think you've seen mucus there. Important in lubricating food. But we've also seen a number of digestive enzymes there, like ptyalin. Ptyalin is the salivary amylase, the one that breaks down carbohydrates. Then we have the salivary lipase, the one that breaks down lipids, among other enzymes important in digestion of substances. There are also some chemicals within the saliva which are antibacterial, for example, immunoglobulin A, that is antibody A. We've also seen lysozyme. So basically, they have antibacterial properties. They're able to fight infections. The water within the saliva is important in dissolving the food particles, basically, but that dissolving is also important in increasing the sensitivity of taste buds to food particles. We can say these are the key functions of saliva. Now, in terms of how the salivary glands look like histologically, before I talk about each of them, I want to give you first a template of how glands look like histologically, so that we are going to narrow down to how salivary glands look like. Then we are going to narrow down to how each of the salivary glands look like. That will be much easier. So what I'm going to do first is to just give you a general outline of how glands in general look like histologically. Remember, a gland is a tissue or a group of cells or an organ that produce and release a substance that performs some specific functions in the body. And there are usually two types of glands. We have endocrine glands and we have exocrine glands. Exocrine glands, like the glands we're talking about today, produce body fluids. These fluids are released within a ductal system. and they travel to epithelial surfaces. Those are exocrine glands. Don't confuse them with endocrine glands, which produce hormones into the bloodstream. For most glands, whether endocrine or exocrine, for most glands, they will have parenchymal components, which are the functional components, the part that produce the secretions, but they'll also have a stromal component. The stromal component refers to the connective tissue framework that hold the gland. It could be a capsule that's surrounding the gland. It could be some connective tissue trabeculae that go into the gland and divide it into lobules. Those trabeculae usually contain blood vessels, nerves, and lymphatics, or even the ductal system. Some ductal system are contained within that stromal tissue. So understand, there's a stromal tissue, which is the connective tissue elements of a gland, and the parenchymal component, which is a functional component of a gland. Now, histologically, if you want to identify a gland, there's some things that we use to identify a gland. First, we just want to look at the organization of the parenchyma. And that's the one I want to just talk about generally. I'll not talk about the others. In terms of organization of the parenchyma, we look at how the cells of the parenchyma of that gland are organized. They could be in a cluster form, which means they're all grouped together in one bundle. Or it could be a cord morphology, which means that the cells are forming kind of some strings of cells, like beads of cells. That's a cord morphology. Or it could be follicular morphology, where you have a ring of cells in the periphery, could be simple or stratified. And then at the center, you have some material. That is a follicle or follicular morphology. If the parenchyma of a gland take any of those three morphological patterns, that gland is an endocrine gland as opposed to an exocrine gland. And perhaps let me give you some images. So you tell me, is it a cluster morphology? Is it a cord morphology or follicular morphology? We'll start with the one projected there. Let me call it question 1 for you. So just put in the chart, is it cluster, cord, or follicular, the one projected? All right, I've seen majority are for follicular, and those are the ones who have gotten it right. You can see the ring of cells surrounding a central material. Second one, I'll call that question 2. Again, put in the chat. 30 seconds you have. Is it a cluster, cord, or follicular morphology? Okay, that one, everyone who has answered has gotten it right, apart from one person. That is cluster morphology. And so it goes without saying that the last one there is a cord morphology. This is how cord morphology look like. You can see cells form things like beads or strings of cells. That is cord morphology. So whenever you look at a gland and the cells take any of these 3 morphological patterns, that is an endocrine gland. It produces hormones into the bloodstream, as opposed to it producing body fluids into epithelial surfaces. Well, I don't know whether you are able to identify these glands Let's start with the first one. Now that's an endocrine gland. Which endocrine gland would that be? So you can just put in the chat, which endocrine gland could that be? Let that be question 3 for you. On the first image, which endocrine gland is that? Okay, those who have answered, gotten it right, apart from one person. So the first one is thyroid. How about the second one? Let's go to the second one. I'll call that question 4. In the second one, which endocrine gland is that one? That might be a bit hard, but there's some clues that you can still use. Well, I'm happy one person has gotten it right. So that is basically parotid gland. You can see the cell types: 1, adipocyte, oxyphil cells, clear cells, and principal cells. Okay, the last one. Let me call that question 5. Which endocrine gland is that one? Question 5, the last image. A good number have gotten it right. So that one is actually the zona fasciculata of the adrenal cortex. Right. So you now know how endocrine glands look like. How about exocrine glands, which is our subject matter? And I'm doing this because we are going to look at salivary glands. We're also going to look at the pancreas, and they'll be having those patterns. That's why I want you to understand the baseline before we look into them. Then the histology will be very easy. So in exocrine glands, the glands are usually either in a tubular morphology, alveolar morphology, or acinar morphology. In this image, this is what tubular morphology looks like. So a gland may look like that. If you cut in cross-section, it will appear that way. For acinar morphology, the gland will look like that. You know, usually exocrine glands will have 2 portions: the ductal portion and the secretory portion. The ductal portion seems to be standard for all of them, but the secretory portion is different. For the tubular one, the secretory portion and the ductal portion look similar. There's no ballooning out. And so in cross-section, it will appear just like that, a tube cut in cross-section. For acinar morphology, the secretory portion is expanded, ballooned out, and that also applies to the alveolar morphology. The secretory portions are ballooned out. But for acinar morphology, the cells are prism-like, very tall. And so that makes the lumen of the acinar morphology be very narrow or even non-existent, like you can't see it nicely in light microscope. It appears collapsed most of the time. That is acinar morphology. For alveolar morphology, the cells which line the secretory portion are simple cuboidal cells. And so basically, the cavity of the secretory portion is ballooned out as well. So this is a tubular, this is acinar, and this is alveolar morphology. And this is how they all look like on cross-sectional microscope. So with that in mind, I have some glands here for you. The first one, second one, third one, and fourth one. They all depict the same thing. I want you to write in the chart, are they tubular, alveolar, or acina? They're all the same. These ones in longitudinal profile, the other two in cross-section. So right, are they tubular, alveolar, or acina morphology? Okay, thank you for those who have responded. So these ones are all tubular morphology, as majority of you have indicated. Now let's try this one. Again, they are the same. The first one is a drawing. The other two are actual slides, but they depict the same concept. Are they tubular, alveolar, or acina morphology? All right, good attempt there for those who have responded. So majority of you are right on that one. These are alveolar morphology. We have simple cuboidal epithelium and a wider lumen. So these are alveolar morphology. It therefore goes without saying that what we have last here is acina morphology. And that is the one we are going to talk more about today, the acina exocrine glands. And that is where I wanted you to pick the others first, then you deal with them. Forget about them. The acina morphology are the ones we're going to talk about. Look at those ones. These are acini. You can hardly see the lumen of the acinus because of what I've told you about the prism-like cells that form the secretory portion of the acini. On these particular images, you also pick something, that some acini cells pick up stains nicely, like the first two, and some of them don't pick up stains nicely. That brings us to the third concept that we look for when you're describing a gland. Especially if you notice that the gland is exocrine gland of acina morphology. When you determine that it's an exocrine gland of acina morphology, then we want you to classify the acini. Are they mucus acini? Are they serous acini? Or are they seromucus acini? In the previous image, the ones which take stain deeply are termed serous acini, like the first image. The ones which the acina cells don't take up stains are termed mucus acini. The reason they're not taking up stains is because of the type of conventional stains that we use, which are usually hydrophilic stains. So they're only miscible with water. The dyes that we use, they just stain water-soluble substances. And mucus is not water-soluble. And that's why they don't pick them. They don't stain them. So with that in mind, I want you to be able to identify the first one I'm giving you. How will you classify this one? Is it mucus acinus? Is it serous acinus? Or is it seromucus acinus? That first one. So that is perhaps the 8th question I'm giving you. I'm going to give you many questions today, and I'm telling them for your information. So that one, in the first image, is it serous, mucus, or seromucus? Majority have gotten that right. That is seromucus type. So these ones are the mucus acini, but these ones are the serous acini. So kind of a mixture of the two. Okay, I'll give you another one. That second image. Is it serous? Is it mucus? Or seromucus? Okay, carry on in the chat. All right, I'll close it there. So majority have gotten that right. This is still seromucus type. Now, a serous type will look like that. The acini are predominantly staining, and a mucus type will look like that. The acini are predominantly not staining. I know you can see these other three which are staining, but if you look carefully, those are not acini. They're ductal portions. Remember, an exocrine gland releases its secretions into a ductal system. So those are basically ducts. So with that in mind, therefore, I can now give you the general histological pattern of a major salivary gland. A major salivary gland is usually lobulated. You can see lobules. So there's one lobule there. You can see another lobule there. You can see another lobule there. It's lobulated. And remember, those lobulations are because of the stromal tissue that enter the gland and divide it into smaller parts. A higher magnification of a salivary gland reveals that salivary gland contain acini. Some acini are serous like these ones. Some acini could be mucus like those ones. So they are acini morphology, that's the take-home message. A major salivary gland may have some scattered fat cells, as you can see those ones. Those are not acini, they are adipocytes which are scattered. Of course, a major salivary gland will also have a duct because they are exocrine glands, so they have the ductal portion. So with that in mind, therefore, I want to take you through each of the three major salivary glands. We talk about them and you'll be able to identify them as well histologically. Let's start with the parotid gland. The parotid gland is the largest salivary gland. It lies on the lateral aspect of the face, just near the temporomandibular joint, anterior to your ears, as you can see in that particular image. The duct of the parotid gland is called the parotid duct or the Stenson's duct. That duct opens into the vestibule of the mouth. From our previous classes, you remember what the vestibule of the mouth is. So it opens into the vestibule of the mouth, opposite the second upper molar. You know, the upper molar around the maxillary bone. So opposite the second maxillary molar tooth. The nerve that stimulates it to produce its secretions will be the glossopharyngeal nerve. Now, glossopharyngeal nerve sends parasympathetic stimulation to the parotid gland, which gives instruction for secretion. And remember, autonomic nervous system consists of pre- and postganglionic neurons. So the preganglionic neurons are largely within the glossopharyngeal nerve and another nerve there, which we call lesser petrosal. But then the postganglionic neurons usually hitchhike on branches of trigeminal nerve, branches of mandibular nerve, specifically the auriculotemporal nerve. The ganglion of synapse is what we call the optic ganglion. So if you trace this image, you see the fibers which provide secretomotor innervation to the parotid gland first come from the brainstem within the glossopharyngeal nerve. Then they leave the glossopharyngeal nerve as lesser petrosal nerve, and they're going to synapse somewhere in the optic ganglion. The postganglionic neurons leave the optic ganglion and hitchhike on a branch of mandibular nerve, which we call the auriculotemporal nerve. And that is how the postganglionic neurons reach the parotid gland. So secretomotor innervation to the parotid gland come from glossopharyngeal nerve. Parotid gland produces predominantly serous secretion, predominantly watery secretion. And so this is how the parotid gland looks like histologically. It's covered by a capsule, as we can see there in that particular magnification. In this particular magnification, we can also see ducts which are within the stromal tissue. We call those ducts extralobular ducts. They're outside the lobules. At this magnification, we can see lobulation. There are those white small cells you're seeing, which are adipocytes, but they are best seen at a higher magnification, which I'm now giving you. So in this magnification, we see scattered adipocytes. We also see the lobules better, and that the lobules contain acini. I'm struggling to identify any mucus acini. What we appreciate is that the acini are predominantly serous. We also note that within the lobules, we have ductal structures like that and that and that one. So we have ducts which are inside the lobules. And yes, these white things are not mucus acini, they are adipocytes. This high magnification confirmed to us that the acini are indeed serous. That's a serous acinus. There's another serous acinus. And these ones are scattered adipocytes. That is a duct. So parotid gland contain predominantly serous acini and of course the duct. So that is a low magnification just to capture that. Lobulation, the presence of serous acini, the ducts, scattered adipocytes, and connective tissue covering. Maybe let's say something about the parotid ductal system. There are different types of ducts in a major salivary gland, and I'll use the parotid as an example. There are some ducts which are outside the lobules, like this one. We call them extralobular ducts. That help us to distinguish them from the ducts which are inside the lobules, which we call intralobular ducts, like that one and that one. So the larger ducts are extralobular. They are found within the stromal tissue, but the smaller ducts are within the lobules. Now, these smaller ducts which are within the lobules, the intralobular ducts, are of two types. There are those ducts which are just forming from the acinus, like that one. We call that one intercalated duct. Intercalated duct. Compare that with this one, which is fully formed with some uniform cells. And look at the basal cell, the basal zones of those cells. They form some fine lines, maybe a high magnification of that will demonstrate to you what I'm talking about. Some faint lines on the basal side. That is what we call striation because of the number and regular arrangement of the mitochondria on that side. These ones are called striated ducts. So two types of intralobular ducts, intercalated ducts and striated ducts. The striated ducts have active transportation mechanism that they perform, and that is why they need a lot of mitochondria as well. Okay, that is parotid gland. Let's talk about the sublingual salivary gland. The sublingual salivary gland, as the name suggests, is just below the tongue, and that is where it derives its name. The ducts of sublingual glands are multiple. It's not just one, but multiple ducts, tiny ones. They open in the floor of the mouth. Now, the sublingual gland receives secretomotor nerve supply from facial nerve. Facial nerve gives a branch, which is called the chorda tympani. And this chorda tympani usually join the lingual nerve as it goes to the sublingual gland. Remember, it is still parasympathetic, so we'll still have preganglionic and postganglionic fibers. In this image, we can trace that. So this is facial nerve. Usually it has two roots. The motor root is this one, and the parasympathetic and sensory root is that one, which is sometimes called nervus intermedius. Anyway, we are following the nervus intermedius, so containing preganglionic fibers, preganglionic parasympathetic fibers coming that way. This region you're seeing there is where the cell bodies of facial nerve fibers are usually found, the one that come from the taste buds. So we call the geniculate ganglion. But let's ignore that one because we're not following the taste right now. We are following the parasympathetic fibers. So we are following the dotted lines. That are those ones which go to another one close to the eye. So we are not following that one. We are following this other one. The fibers are here. So they leave the facial nerve to enter into a nerve which we call the chorda tympani nerve. Chorda tympani nerve join a branch of the mandibular nerve, which we call the lingual nerve. This branch of mandibular nerve called lingual nerve is going to the tongue. So chorda tympani joins the lingual nerve, but the fibers are still running there. Then they'll synapse there at the submandibular ganglion, which this book is also calling the submaxillary ganglion. Let's call it submandibular ganglion. From there, postganglionic fibers go to both sublingual gland as well as submandibular gland. So the same pattern of innervation, route of innervation from submandibular is the same route of innervation for sublingual. The sublingual gland produces predominantly mucoid saliva. And so we'll expect the sublingual salivary gland to be predominantly mucus acini. As we can see in this image, we see a gland with predominantly mucoid acini. That is what we expect for the sublingual salivary gland, predominantly mucoid. The third one, submandibular gland, located below the mandible. The duct of the submandibular gland is known as the Wharton's duct. It drains to the floor of the mouth via some tiny openings below the tongue, which we call the sublingual puncta. Now, in this image of the mouth, we can see this one, which we call the lingual frenulum. On either side of the lingual frenulum, we can see the impressions formed by the sublingual gland. So that is the location of the sublingual gland, even that one. But just medial to that, we can see the openings of the duct of the submandibular gland. So those are the regions we call the sublingual puncta. Maybe this image is better in showing you the submandibular duct and its position of opening on either side of the lingual frenulum. Remember, the fold down there is formed by both the submandibular duct as well as the sublingual gland. So in terms of nerve supply, similar to the innervation to the sublingual gland, the pathway is that long one that I just described. Submandibular gland produces predominantly mucus saliva, which means it will have both mucoid as well as serous components. And so histologically, we'll expect the parenchyma of the submandibular gland to be of mixed acini. We can see serous acini and we can see mucus acini. In addition to, of course, us appreciating the ductal portions, as we can see there. This image captures something very important about the acini. Yes, we have mucus acini and we have serous acini as well. There's some regions, look at that mucus acinus. There's some regions where the serous cells make something like a crescent moon around the mucus acinus. This crescent moon-like structure of a serous cell is termed serous demilune. The submandibular gland contains serous demiliones. So this is an arrangement where you have a serous cell just embracing the mucus acinus. Serous demiliones are unique to submandibular glands. So because of the presence of mucus and serous acini, the submandibular gland produces mixed saliva. Good, now we're done with the salivary glands. We can pick the next organ, the liver. In terms of gross morphology, we can talk about the location first. The liver lies within the right hypochondrium as well as within the epigastrium. I believe you are already aware which part of the abdominal cavity are we calling the right hypochondrium and which part are we calling the epigastrium. The liver is below the right hemidiaphragm, just below the right hemidiaphragm, and also below the central tendon of the diaphragm. The central tendon of the diaphragm is where the heart also rests. So heart on top, then diaphragm, then below that one, the liver. So that means that the liver extends up to the midline generally. We describe the liver as having two surfaces, the surface facing the diaphragm and the surface facing the abdominal viscera. The surface facing the diaphragm is called the diaphragmatic surface, and that's the one that is convex and smooth, as we can see here. So this is the one that will be lined by the right hemidiaphragmatic dome as well as the central tendon of the diaphragm. Then we have the visceral surface, which is this side. The visceral surface is relatively irregular and it is related to abdominal visceral organs. Many of them make the impression on the visceral surface of the liver. In this image, we are seeing the diaphragmatic surface there. And then the diaphragmatic surface usually have a ligament there, which we call the falciform ligament, that tend to separate the right and left anatomical lobes of the liver. So that's the falciform ligament that attaches to the anterior abdominal wall. The visceral surface of the liver, as I've told you, is irregular. This is the visceral surface. Related to a number of abdominal organs, we can mention a few. So this is where the stomach makes the impression. That is where the esophagus makes its impression. We also have impression for the right kidney somewhere there, impression for the colon there. Of course, the impression for the inferior, so for the gallbladder is there. Actually, the gallbladder is still intact on this image. We usually also have impression of the inferior vena cava somewhere there. The duodenum will make an impression somewhere around here. Apart from that, I want us to talk about the lobes of the liver. So we consider the liver as having right and left anatomical lobes, as well as right and left physiological lobes. Let's define the anatomical lobes. So the right and left anatomical lobes are separated by the falciform ligament. When you look at the diaphragmatic surface, DS stands for diaphragmatic surface. We have the right and the left anatomical lobe of the liver. This is a falciform ligament. However, if you look at the visceral surface of the liver, we will note that it's separated by something else we are going to see. So there's what we call ligamentum venosus of the liver. This is also what we call the ligamentum teres of the liver. Well, you can call it the round ligament of the liver. So on the visceral surface, we are going to note those ones, still separating the right and left anatomical lobe. But let's first talk about the right and left physiological lobe. The right and left physiological lobes of the liver are based on blood supply or venous drainage. And so the right and left physiological lobes correspond to the plane of the middle hepatic vein. The liver is drained largely by three major veins: right, middle, and left hepatic vein. The plane of the middle hepatic vein separates the right and left physiological lobe. But because we can't see that from inside, from outside, what do we see? From outside, what we see is that it is corresponding with the impressions of the gallbladder as well as impressions of the inferior vena cava. So here, this line here that you see there, that imaginary line, is a line separating the right and left physiological lobe. The plane of the middle hepatic vein correspond with the impression of the gallbladder here and the impression of the inferior vena cava there. So this is the plane of the middle hepatic vein. So this one will be right lobe and this one will be left lobe. This one has been rotated. So we're looking at the liver from behind. So this is still left and this is right. Physiological lobes. If we are to look at it from anatomical perspective, then this is the impression of the ligamentum venosus. And this is the region for the round ligament or ligamentum teres of the liver, which correspond with the location of the falciform ligament as well, if you are to look at the diaphragmatic surface. So anatomically, this is the plane of separation of the left anatomical lobe and the right anatomical lobe. Physiologically, this is the plane of separation of the left physiological lobe and the right physiological lobe. So the take-home message is what? That the physiological lobe and the anatomical lobes have a different point of separation. The plane of separating them is very different. In this image, you can see that that contentious issues here, and this region is the contentious issue. You can see it's written there, the caudate lobe and the quadrate lobe of the liver. Anatomically, the caudate and the quadrate lobes of the liver are on the right anatomical lobe, but physiologically, they are on the left physiological lobe. So if you look at the right and left physiological lobes, caudate and quadrate are on the left, but anatomically, they're on the right. That's a take-home message from that long description. And that you also see here. So the plane of the anatomical separation is there. Caudate and quadrate will be on the right. The plane of physiological separation is here. Caudate and quadrate will be on the left. This point where several blood neurovascular structures enter the liver is known as the portal hepatis. At the portal hepatis, you have the portal vein. We also have the hepatic artery, and we have the common bile duct as well. Let's talk about the blood supply to the liver then. The liver receives two vascular systems. We have the hepatic artery which is a branch of the celiac trunk, contributing about 20-25% of blood flow to the liver. That vessel delivers oxygen to the liver. But we also have the portal vein, which delivers quite a chunk of blood, 75 to 80% of blood flow. But remember, portal vein is coming from the digestive system, so it is carrying deoxygenated blood. However, it contains nutrients that is in the blood that was running through the intestines, so that blood absorbs nutrients from the intestines. Unfortunately, also, if there are some toxins that are in the food that you've taken in, those ones are also within that particular blood, and that is directed to the liver. So this image captures for you the fact that there are two sources of blood supply, the portal system and the hepatic artery. But either way, there's mixing within the liver. Then from the liver, we have the three hepatic veins, the right hepatic vein, middle hepatic vein, and left hepatic vein, taking blood directly to the inferior vena cava. So that's the liver blood supply. We talked about splanchnic circulation in our previous class. I think that one is not hard. Now let's describe something slightly hard based on the blood supply to the liver. And that is the segmental anatomy of the liver. The liver is described to have, yes, two functional lobes, right and left, but those functional lobes of the liver are further divided into segments based on the blood supply to the liver. We have a total of 9 segments in the liver. The person who described them goes by that name, Conan segments. Now, how do we get the 9? It may appear hard, but let's just follow together. It's not hard. We know that portal vein enters the liver from the intestines through the porta hepatis. When it enters, it divides into 2 major branches, the right and the left branch of the portal vein. The plane of division of the portal vein into those 2 is relatively horizontal, passing through the liver. And so that plane divides the liver into 2 portions, the superior portion and the inferior portion. Let's start there. So these ones are the superior portions, and these ones are in the inferior portions. If you've understood that, then the rest is easy. So there are those segments which are on above that plane, and there are those segments which are below that plane. So how do we go about it? Let's first define what is segment 1. Segment 1 of the liver is the quadrate lobe. That lobe just next to the inferior vena cava. From the previous image, you saw it nicely. That lobe that is contentious. Is it on the right or on the left? So anatomically on the right, but physiologically on the left. That becomes the segment 1. It is more posterior. Segment 2 of the liver. Okay, before I go to segment 2, let me put it this way. After we've divided the liver into right and left functional lobes, look at the plane of the portal of the hepatic veins. The middle hepatic vein is there, which divides the liver into right and left physiological lobes. This is the middle hepatic vein, dividing the liver into right and left physiological lobes. Then we have the right hepatic vein, which will be on the right physiological lobe, and the left hepatic vein, which will be the left physiological lobe. The right and the left hepatic veins further divide their corresponding physiological lobes into two portions. So the left hepatic vein divide the left physiological lobe into a medial segment and a lateral segment. However, the right hepatic vein divides the right physiological lobe into anterior segments and posterior segments. So if you've gotten that division, and remember the plane of the portal vein split the liver into upper segments and lower segments. Now we are good to go in knowing segment 2 to 8, and I'll tell you why I said there are 9. So segment 2 is basically the left lobe, which is on the superior aspect and lateral as well. So the left lateral superior, let's call it the superior left lateral, or the left superior lateral segment. You can call it the left lateral superior segment. Segment 3, the left lateral inferior segment. Segment 4 is the left medial segment, but segment 4 has 2 parts. We have 4A and 4B. That's what makes them 9. So 4A is superior and 4B is inferior. Segment 5 is this one here. So we are on the right, we are also below, and we are anterior. So the right anterior inferior segment. Segment 6, the right posterior inferior segment. Segment 7, the right posterior superior segment. And finally, segment 8, the right anterior superior segment. I hope you've seen that I've gone in an anti-clockwise manner. Okay, that was clockwise, but if I was to look at it from behind, it will be anti-clockwise. Anyway, so those are the liver segments, a total of 9 segments. You will have to go through that again at your own time for you to understand them. But this also captures the liver segments nicely. So the left superior lateral is 2, left inferior lateral is 3, the left superior medial is 4A, left inferior medial is 4B. We have the right anterior inferior is 5, right inferior posterior is 6, right superior posterior is 7, and right anterior superior is 8. Remember, this is the plane of the middle hepatic vein. That Cantley's line is the plane of the middle hepatic vein. This one here will actually be the plane of the falciform ligament, which separates the right and the left anatomical lobes, but it's also the plane of the left hepatic vein. Then this one is the plane of the right hepatic vein, dividing into anterior and posterior segments. The superior and inferior segments are divided by the plane of the portal vein, splitting into 2, upper and lower parts. Remember, the right physiological lobe is divided into anterior and posterior, but the left physiological lobe is divided into medial and lateral. Let's see why we are actually learning the segmental anatomy of the liver. I'm showing you some CT scan images of the abdomen. And I want you to notice the liver. So there are two images that I'm going to run concurrently, the one on your left and the one on your right. The one on your left has bright arteries. So there's a drug that is usually given, and that image is acquired when the drug is still within the arteries. So we call the arterial phase. The one on your right has the veins being opacified, and specifically the portal vein having a lot of that drug. So we call the one on your right the portovenous phase. This organ here is the liver. And on the left side, this organ here is the liver. So they're still playing. That organ there is the liver. This organ is the liver. So I want us to then use this to help us determine the segments of the liver. Now, this is the kidney, which means we are on the lower parts. So the segment that is next to the kidney, that will be segment 6. This is segment 6 of the liver. That is also segment 6 of the liver. As opposed to if I gave you another segment, look at that one. So in the lower zones, and you can see the falciform there. So this one is most likely segment 3 of the liver. We're in the lower zones and we are lateral to the falciform. So that is segment 3 of the liver. So we usually use the segmental anatomy to know the location of different pathologies within the liver. And so when you get reports of there's a lesion in segment whatever, you should be able to pinpoint and place that segment. That's why we learn the segmental anatomy of the liver. Histologically, this is how the liver looks like. So we see a lot of polygonal structures. The one for the pig, which is the one I'm showing you right now, has a lot of connective tissue splitting it. But the one for humans don't have those ones. But either way, so the one we are seeing right now is human. Either way, okay, it's not from pig. It may not be human. It looks a bit small for human, but it's not from pig. Maybe from a rabbit or something. So what we see, we still see some polygonal structures there, even that one. And I want to look keenly at the center of this polygonal structure, and we see some large vessels. The vessel at the center of the polygonal structure is known as the central vein of the liver. So each polygonal structure usually has that central vein, even though you don't see in all of them. Higher magnification reveals the presence of the central vein within each polygonal structures. Those polygonal structures are roughly 5 to 6-sided. And usually they're 6-sided, except that when we capture in the microscope, we may not be able to see the 6, but they're hexagonal in nature. Each hexagonal structure has a central vein at the center. The corners of the hexagon may contain something else, like that one. The structures which are contained at the corners of the hexagon are known as the portal triad. A portal triad consists of 3 things: a branch from the hepatic arteriole, a branch, so you can just call it hepatic arteriole, a branch from hepatic artery, a branch from the portal vein, which we call portal venule, and a tributary of the bile duct. We can call it bile ductule. So portal triad contain 3 structures: hepatic arteriole, portal venule, and the bile ductule. So those ones, the vessels bring blood to the liver and pass through the liver cells and drain to the central vein. Then the central vein are the ones that join to form the hepatic veins that take blood out of the liver. So this is how it looks like, that magnification. And this higher magnification revealed that the cells of the liver are even arranged in cords as well. As you can see, these are cord arrangement of the liver cells. That's a central vein, and this is what we call a portal triad within this region. We have a portal, venule, hepatic, arterial, and bile ductal. The cells of the liver are anchored by some fine connective tissue, collagen type 3, what we call the reticular fibers. And what you're seeing here is a specific stain that picks the reticular fibers, providing structural support to each and every cell type of the liver. What are the cell types of the liver? The most abundant of the cell type are called the hepatocytes. They're the ones that perform most of the liver functions. Apart from hepatocytes, we have the Kupffer cells, which are basically macrophages within the liver. We have hepatic stellate cells of Ito, which metabolize vitamin A. They also store fat. We have endothelial cells, which line the capillaries within the liver. These gaps you're seeing here are actually capillaries. We call them sinusoids. They line the liver sinusoids. Those are endothelial cells. And lastly, we have the cholangiocytes. The cholangiocytes are the epithelial cells that line the bile ductules, which are within the portal triads. So cholangiocytes will line the bile ductules. These are the cell types of the liver. Based on that, let's describe the histological lobules of the liver. I showed you some hexagonal structures. A hexagon will have 6 corners and 6 sides. The center of the hexagon contains the central vein. The corners of the hexagon contain the portal triad. A portal triad has 3 structures: hepatic arterial, portal venule, and bile ductal. So that's a portal triad, that's another portal triad, that's another portal triad. Based on the arrangement of the hepatocytes and those blood vessels, we describe 3 histological lobules of the liver. The first one is what we call the classical lobule of the liver. The classical lobule of the liver is a hexagonal structure that marks the territory drained by a single central vein. Blood comes through the portal venule and hepatic arterial and flow toward the central vein, that way or that way, this way or that way or that way. So the territory drained by a single portal vein is called the classical liver lobule. Another one is what we call the portal lobule. The portal lobule is a rectangular lobule which marks the territory supplied by a single portal triad. So you can see the portal triad is at the center, and the corners are the central veins that defines the portal lobule, as opposed to the classical lobule where the corners are the portal triads and there are 6, and the central vein is 1 at the center. For the portal lobule, 1 portal triad at the center, and the corners have central veins. Those 2 are largely histological. The last one is called the hepatic sinus. Hepatic sinus is considered the functional lobule of the liver. And this one is based on blood flow through the liver. Now let's look at how blood flows again. So from this one, blood flows that way, or that way, or that way. And from this one, blood flows that way, or that way, or that way, if it was there. That helps us to define a diamond. A diamond has 4 corners. The 2 corners opposite corners containing central veins, and 2 opposite corners having the portal tract. And that's what you see there. So it's not a structural entity per se, it's more of a functional unit rather than a structural entity. And that is why we are calling the functional lobule of the liver. It's called the hepatic sinus. It's based on blood flow. It's a diamond in shape, as you can see. Now, based on the hepatic sinus, which is the functional lobule of the liver, it helps us to define 3 hepatocyte zones within the hepatic sinus. Look at it again. Blood from the portal vein enter like that or like this. So the cells in this zone will receive the blood first, followed by this zone, and lastly that zone. So zone 1 represents the population of hepatocytes that receive blood first, either from the portal venule or from the hepatic arteriole. And zone 3 contains the population of hepatocytes that receive blood last, either from the portal venule or from the hepatic arteriole. Those have implications, and I want to think through the clinical implications of those ones. Suppose there is some toxins from the GIT, which zone will be affected first? And how about the cells which are very active because they receive a lot of oxygen? Is it zone 1, zone 2, or zone 3? Suppose you have some congestion in the inferior vena cava, which ones will be affected first? Suppose there's some ischemia, which ones will be affected first? Zone 1, 2, or 3? Think about those ones. So in this image, we see a portal tract there and the central vein. So blood will be flowing that direction. Therefore, the hepatocytes here are in zone 1, then zone 2, and then zone 3. That is liver histology. Let's talk about metabolic functions of the liver. So there are a lot of metabolic functions of the liver. We start with carbohydrate metabolism. The liver functions as a glucose buffer because they're able to store extra glucose in form of glycogen, and when you need it, it can break down that glycogen. It is also important in converting galactose and fructose to glucose. The liver also can form glucose from non-carbohydrate substrate, from non-carbohydrate precursors through the process of gluconeogenesis. And there are many other compounds that are formed from the intermediate forms of carbohydrate metabolism within the liver. How about as with regard to fat metabolism? The liver is able to oxidize fatty acids to give us energy. The liver also synthesizes some types of lipids like cholesterol, phospholipids, which go to cell membranes, and cholesterol, remember, also goes to bile, and lipoproteins, basically, that help in transport of substances. The liver can also synthesize fat from non-fat precursors like proteins and carbohydrates. Then after it has synthesized, these ones then go to adipose tissues for storage. That's why you're told if you eat a lot of carbohydrate, again, you're still going to add weight because the carbohydrate is going to be converted to fat, and that fat is going to adipose tissue for storage. The liver also metabolizes proteins. We talk about deamination of proteins, which is an important step that must happen before we can use these ones to provide energy. We also have formation of urea. These are mechanisms that the body uses to eliminate ammonia from the body fluids. We have plasma proteins which are formed by the liver, and basically the liver forms almost all types of plasma proteins apart from the gamma globulins, which are usually part of antibodies, and so they're usually synthesized by the plasma cells. There are many other conversions that occur in the liver for the amino acids, so one amino acid to another, but also the liver is important in synthesis of the non-essential amino acids, you know, those, the ones that your body is not able, you have those amino acids that your body is able to synthesize, and those amino acids that you can only get from plants. So the liver can synthesize some types of amino acids. Apart from metabolic functions on carbohydrate, proteins, and lipids, we know that the liver also stores some vitamins, A, D, and B12. It's also acting like a buffer for iron because it's able to store iron in form of what we call ferritin. And this apoferritin system is an important, ferritin system is an important one because basically when you have a lot of iron, it binds with apoferritin in the liver to form ferritin. And when you need iron, it releases, the ferritin releases the iron, then you remain in apoferritin. So apoferritin is what is in the liver. Ferritin is the storage form of the iron. We have synthesis of vitamin K dependent coagulation factors, and we also have excretion of various substances through bile juice. And you can excrete a number of things, drugs, some hormones like thyroid hormones. And calcium is also excreted actually primarily through bile. Apart from metabolic functions, there are still other functions like immune functions of the liver because of the presence of the Kupffer cells. And the liver can hold quite a large amount of blood. And so that helps it to be a reservoir for blood. Good, that is the liver. We have two more, but those ones are very quick, so we'll finish them. Let's now talk about the biliary system. So before we talk about biliary system, just to make sure that we are still awake, let's try to identify the structures labeled A to D. Let's start with A. So that should be our question 10. What is structure A? Okay, that's a good one. Everyone has gotten that right. Let's identify then structure D. So what is structure D? Now we're in structure D, dog, D for dog. Okay, everyone has gotten that right, apart from one person who has said portal vein. Okay, you've changed your mind. Good. I was getting worried. Someone has said celiac artery. Another one is saying sphincter of Oddi. I think you're hungry now, but okay, we're almost finishing. So A is the liver, B is the gallbladder, C is the pancreas, and D is the duodenum. So we've identified those four. Now, let's talk about the parts of the biliary tree. When hepatocytes synthesize bile, the bile enters through some channels which we call bile canaliculi. And these bile canaliculi drain the bile to the bile ducts which are within the liver. We call them intrahepatic bile ducts. So let's consider this to be the intrahepatic bile ducts, the ones which are found within the portal triads. The ones from the right and the ones from the left join eventually to form what we call the right and left hepatic ducts. The right and left hepatic duct then join to form what we call the common hepatic duct. So I hope you're following. We had intrahepatic ducts, now right and left duct, and now common hepatic duct. The common hepatic duct is joined by the duct from the gallbladder. The duct from the gallbladder is called the cystic duct. These two join to form what we call the common bile duct, commonly named the CBD. The common bile duct is joined by the duct from the pancreas, which we call the main pancreatic duct. And that main pancreatic duct, after joining the common bile duct, they don't form another common channel, they just open together. The point of opening, which I'm pointing at right now, is called the ampulla of Vater. It is in the duodenum. We also call it the major duodenal papilla. So that's the duodenum, and that's the major duodenal papilla, or the ampulla of Vater. Around that opening, we have a sphincter, and that sphincter is called the sphincter of Oddi. So that is basically the biliary tree. Histologically, this is how the gallbladder looks like. It has three major important layers: the mucosa, the muscular layer, and the adventitia. The adventitia is loose connective tissue, as you can see. It may have some fat, but fine, it is the adventitia. The muscular layer contains smooth muscle, very thick layer of smooth muscle running in different directions. The mucosa of the gallbladder is highly folded mucosa. The foldings are so that when it stretches, when it's accommodating bile, it can stretch, it can allow for stretch. That's the importance of the folds. The importance of the folds is to increase the surface area for absorption of substances. The gallbladder concentrates bile. We can see the lining of the epithelium is simple columnar epithelium in that particular image, simple columnar epithelium. This high magnification revealed that the epithelium actually has microvilli on its surface. You can see that thing that looks like a brush border. That is microvilli. So it's lined by a simple columnar epithelium with microvilli. That is an absorptive epithelium. Very important. The muscular layer, as you can see, contains smooth muscles running in different directions. Look at the direction of that one, the direction of this one, this one, this one, different directions. Remember, this one helps it to contract in those all directions. And I told you the outermost layer is the adventitia, which is a loose connective tissue. So while we're on the biliary tree, let's talk about bile juice. Bile juice is secreted both by the hepatocytes as well as by the cholangiocytes. So what secretes what? The hepatocytes secretes the organic components of the bile juice, and that is released into the bile canaliculi. As the bile canaliculi join to form the bile ducts, bile ductules and bile ducts, the bile ductules are lined by cholangiocytes. The cholangiocytes produce watery solution. That watery solution contains now the inorganic components of bile juice. So two cell types produce bile, hepatocytes and cholangiocytes. The quantity of bile secreted depends on a number of things. Two major factors determine the quantity of bile produced. The availability of bile salts. Usually bile salts circulate in the bloodstream. They go to the liver. From the intestine, they're taken back to the liver. We're going to talk about what we call enterohepatic circulation. And is one of the gut hormones. Secretin acts on the cholangiocytes, and it promotes the secretion of that watery solution that contain inorganic components. The main inorganic component that we're talking about here is bicarbonate. So that bicarbonate-rich solution comes from the cholangiocytes under the stimulation of secretin. Now, when bile is formed, bile will go to the gallbladder for storage. But during storage, bile is also concentrated. So the gallbladder will also concentrate the bile. And when the time comes that that bile is required for digestion, gallbladder contract to release the bile. So we usually say the gallbladder has 3 key functions: storage of bile, concentration of bile, and release of bile during digestion. As you think through that, I want you to then think about the possible routes that bile will take. When you are not eating, bile will come from the liver, through the hepatic ducts, common hepatic ducts, cystic duct to the gallbladder for storage when you're not eating. But during mealtime, bile comes from the gallbladder. It contracts. So bile will go to cystic duct, then common bile duct, then ampulovator. But bile can also come from the liver direct and follow that route all the way to the duodenum. 3 possible routes of bile flow. What's the mechanism of secretion of bile? The presence of fatty acids within the duodenum will induce the duodenum to produce cholecystokinin. Cholecystokinin in turn causes gallbladder to contract, but it also causes the sphincter of Oddi to relax. So it's a combined effort, relaxing the sphincter of Oddi and contracting the gallbladder. Gallbladder contraction is also stimulated by the cholinergic stimulation, which comes from either the vagus nerve, parasympathetic, or the enteric nervous system, the ones that release still acetylcholine as a neurotransmitter. So it's a cholinergic response either from vagus nerve direct or from the enteric nerve terminals. We can produce about 600 to 1,000 ml of bile per day. That can also vary actually, depending on whether you're eating or not eating. And if you're eating, what are you eating? Fatty meal will make you produce more bile. Bile contain a lot of water fine. The solid component can be classified into organic and inorganic. The inorganic ones, you can see quite a number, but importantly, I want you to pick calcium, which is excreted through bile. The major form in which the body excretes calcium is bile, but also bicarbonate. And that bicarbonate is important in neutralizing the chyme from the stomach. As with regard to the organic components, we have bile salts, which are usually from the circulation, but also formed by the liver from cholesterol, and bile pigments. The bile pigments here refer to the remnants of heme. When red blood cells are broken down, hemoglobin is broken down into heme and globulin. The heme component, you take away iron, and what remains, bilirubin and biliverdin, are what you're talking about here. So bile pigments are those remnants of hemoglobin breakdown. Very important. But yes, we also have cholesterol. This is a mechanism of excreting extra cholesterol from the body. We also have fatty acids and others. But importantly, remember, bile salts and pigments are present in bile. You can look at that later, look at the concentration of various substances when they leave the liver vis-a-vis when they leave the gallbladder. And the point to note here is that the gallbladder performs concentration. So the gallbladder will take back some water. That's why the concentration of water is reduced. But importantly, the salts will be concentrated. That's why the concentration of bile salts have increased. Also, the concentration of bile pigments will have increased. That also applies to the concentration of the cholesterol. So there's some things which increase their concentration, and importantly, look at the calcium as well. That's significantly increased because of the concentration function. Bicarbonate reduces because a lot of that is taken back when water is taken back. Right. So what are the functions of bile? The bile salts have a role in fat digestion and absorption. In particular, bile salts act like a detergent. So they break down large fat particles into smaller particles, increasing their surface area, over which now enzymes can act on. But other than that, once the enzymes have acted on end products, on fat, the end products of fat are still surrounded by bile salts, forming what we call micelles, which can now be easily absorbed by the cell membrane of intestinal epithelium. Apart from that, bile is also a mechanism of excretion of various substances from the bloodstream, like bilirubin, the end product of hemoglobin destruction. Those are the bile pigments. Extra cholesterol, excess calcium, even some drugs are also excreted through bile. And lastly, the bicarbonate that is contained within bile help to neutralize gastric acid. Remember, chyme from the stomach is acidic, so we need to neutralize that one. Now, there are many mechanisms for neutralizing it. Bile is one of them. Pancreatic juice is another one. The secretions from the crypt of liver cone, as well as the secretions from the Brunner's glands, they all aim at neutralizing gastric acid. Enterohepatic circulation referred to the circulation of bile salts between the liver and the intestines and back to the liver. So this is the story. The liver produces cholesterol, and that cholesterol is what's used to form the bile salts. The bile salts go through the biliary tree. There'll be concentration fine, but eventually they find themselves in the duodenum. So they are useful in the duodenum for absorption of fat, or rather digestion of fat and absorption of fat. About 95% of those bile salts are actually reabsorbed back, either through passive transport in the proximal small bowel or active transport in the distal small bowel, but they all go back to the bloodstream. So when they go back, they go to the portal circulation. 95% is reabsorbed. Only 5% go to the stool for excretion. The 95% that is reabsorbed enters the liver again. And that is what I was talking about. Availability of bile salts will help the liver to produce more bile. So as long as that one is there, it will produce more bile. So that 95% is added to another extra 5%. So at any given time, the liver will be producing some other extra 5% from cholesterol. The point here is that the liver, the body requires these bile salts for absorption, for digestion and absorption of fat. And so they are usually recycled. This is the enterohepatic circulation. So it's about the return of bile salts back. We can interchange the term bile salt with bile acids. You use the term bile acids when you have the acidic components of them. All right, that is the story of biliary tree. Now let's finish with the pancreas. We're going to look at the gross parts, histological structure, and functions of the pancreas. In terms of location, the pancreas is located within the epigastrium as well as the left hypochondrium, usually around the curvature of the duodenum. Behind the stomach, and that region is what we call the stomach bed. We talked about the stomach bed in the first class. The pancreas is usually slightly slanted. The tail of the pancreas is slightly upper level compared to the head. So usually it slants like this, slightly. So that if you are to take cross-section imaging of the body, from above, we'll be slicing the tail of the pancreas before we slice the head of the pancreas. There are 5 anatomical parts. This is the tail, which is usually in the left hypochondrium. This is the body, which is the largest part. The neck is this region sandwiched between the superior mesenteric vessels and the celiac trunk. So that is the neck of the pancreas. The head of the pancreas, that region, medial to the neck, largely around the curvature of the duodenum. The lower part of the head of the pancreas is what we call the uncinate process. And we look at how it comes about embryologically. In terms of function, the pancreas has both endocrine and exocrine components. The exocrine component releases secretions into the ductal system. As we said when we're starting, the endocrine component releases secretions into the bloodstream. Let's talk about the exocrine pancreas. The exocrine pancreas consists of several serous acini, which are found all over the pancreas. Now you know the meaning of acini, you know the meaning of serous. There are several serous acini found all over the pancreas. So this is one acinus, that's another acinus. They are found all over the pancreas. These are the ones that produce pancreatic juice into the ductal system that eventually is transported to the duodenum. The pancreatic acini constitute about 70% of the total pancreatic mass. The principal cell types within the acini are what we call the acinous cells, which are here. Centroacinous cells, I'm going to show you shortly, and the ductal cells. The ductal cells are the cells that line the duct of the exocrine pancreas. So this is histology image of the pancreas, showing you a lobulated organ. We can see some thin connective tissue surrounding it, but we can see multiple lobulations, different sizes. We can also see ductal structures, very prominent ductal structures, telling us that this organ is exocrine, or at least has exocrine functions. A higher magnification of one lobule confirms the presence of lobulations. Also some blood vessels we can now see. But it also captures something very interesting. We have some cluster of cells. Remember, when you talk of cluster, you think endocrine. We have some clusters of cells, light staining, but they're scattered within the acini. These are the acini. And look at the acini. They are predominantly serous acini. They're staining. So we have predominantly serous acini and scattered clusters of light staining cells. The acini are the pancreatic acini. The clusters here are the islets of Langerhans. The cell types of the acini are 2 types. There are those ones which are in the periphery, we call them the acinous cells, and then there are those ones which are at the center. We call them centroacinous cells. The centroacinous cells represents the initial segment of the pancreatic ductal system. The ducts of the pancreas begin from the inside of the acini. And so in cross-section imaging, they'll appear like cells at the center of the acini. We call them centroacinous cells, but basically they're ductal cells. Okay, with that in mind, let's then talk about the pancreatic juice produced by both the acinous cells and the ductal cells. The acinous cells produce the digestive enzymes, different of them, many enzymes. The ones that break down proteins, the one that break down carbohydrates, the one that break down lipids. And for the proteins, the one that inhibit some of them, they're produced by the acinous cells. The ductal cells produce a bicarbonate solution, a sodium bicarbonate solution. So it means it has water and sodium bicarbonate in solution. So those ones are produced by the ductal cells. The small duct, the large duct, they produce bicarbonate. The key stimulants for production of pancreatic juice are three. Cholinergic stimulation, which is either from the vagus nerve or enteric nervous system, produce their stimuli, and that stimuli act largely on the acinar cells for secretion of digestive enzymes. Cholecystokinin also stimulates the acinar cells to produce digestive enzymes. But secretin acts on the ductal cells, and it helps to promote the secretion of the bicarbonate solution. So water and bicarbonate. That image captures for you the stimuli as I've already described. We have the one for vagus, and we have the one from secretin and cholecystokinin. We had already talked about cholecystokinin and secretin in our previous class. Pancreatic juice usually flow through the pancreatic ductal system all the way to the duodenum, specifically in the second segment of the duodenum, the D2. There are two ducts actually from the pancreas. The main pancreatic duct, which is also called the duct of Wissang, is usually joined by the common bile duct, and they both open together at the duodenal, the major duodenal papilla, or what we've called the ampulla of Vater. So this is the ampulla of Vater, which receives both the main pancreatic duct and the common bile duct. The main pancreatic duct drains the tail of the pancreas, the body of the pancreas, the neck of the pancreas, and the lower part of the head of the pancreas. But there's another duct, which we call the accessory pancreatic duct. It's a variant one, which means sometimes not there. However, when present, the accessory pancreatic duct empties into the minor duodenal papilla, slightly up. It drains the upper part of the head of the pancreas, upper part of the head of the pancreas. It's also called the duct of Santorini. Pancreatic juice contain water fine, but also contain some solids. The solids could be inorganic components, and remember, bicarbonate is a good one to mention in that particular list. The organic components are many, but importantly, remember they are digestive enzymes, largely. Well, apart from that, we also have other organic components which are not necessarily enzymatic, but enzymes are the major thing. What are the functions of the pancreatic juice? The bicarbonate helps to neutralize gastric acid. The digestive enzymes are important for digestion. Importantly, the digestive enzymes which break down protein, we call them proteases. The proteases are produced in their inactive form, in their inactive state. Why is that important? So that they don't digest the pancreas itself, or even the intestines, they don't digest. So the pancreatic enzymes that enter the duodenum, we are looking at the ones that break down proteins. These are the ones we call zymogens. They are in their inactive form. When they come, they must be activated. So one of the enzymes which come from the pancreas is called trypsinogen. It is the main enzyme that breaks down proteins, but it comes in its inactive form. That trypsinogen is usually produced together with something called trypsinogen inhibitor. Sorry, trypsin inhibitor, which is also released by the acinar cells. So that's what makes this enzyme not to be activated within the pancreas until it reaches the intestines, where it can be activated to trypsin. Once it has been activated to trypsin, the trypsin is the one that activates the other enzymes, which are important for digestion. So it's a protective mechanism. But trypsin itself also is important in digestion of proteins as well. So it also helps in digestion of proteins. It's an active enzyme. And the second image captures that flowchart very well. So the point here is the pancreatic enzymes are produced in the inactive state so that they cannot auto-digest the pancreas itself. In cases where we have obstruction of the pancreatic duct, we are relying solely on trypsin inhibitor. In that case, the pool of secretions there might be overwhelming the trypsin inhibitor, and so the trypsinogen become activated trypsin. Then they are chaotic. That will cause pancreatitis. Acute pancreatitis is a life-threatening thing. Why? Because when these enzymes are activated within the pancreas, they'll auto-digest the pancreas, and they can also leak into the circulation. You can imagine what kind of shock they're going to cause in the whole body. Great, that was exocrine pancreas. Now, endocrine pancreas, we're almost finishing. The endocrine pancreas consists of clusters of scattered hormone-producing cells, which we call the islets of Langerhans, if you remember from our lecture on endocrinology. So this is an islet of Langerhans, that's another islet of Langerhans. The islets are usually richly vascularized, as you can see blood vessels very near them, because they release their secretions into the bloodstream. They are found all over the pancreas, but most of them are found within the pancreatic tail. They constitute about 2% of the total pancreatic mass, but there are many, close to 2 million of islets within the whole pancreas. I know you're wondering, so the pancreatic acini contributed 70%, this one contribute 2%. How do you account for 28%? Think about the connective tissue elements, think about the ductal elements of the pancreas. There are different cell types which are within the islets of Langerhans, and I hope you do remember them from previous class. We have alpha cells, beta cells, delta cells, and the PP cells, the major ones. Histologically, I've already shown you this slide. So these are the islets of Langerhans, clusters of hormone-producing cells. These are the islets of Langerhans. So remember the secretions, glucagon is one of them, which come from the alpha cells, also called the A cells, help in elevating sugar levels. Insulin come from the beta cells, which are also called the B cells, help to lower sugar levels. And we talked about the mechanism that insulin uses to lower sugar levels from previous class. You can remember that aspect. Somatostatin come from the D cells. This somatostatin is similar to the somatostatin that we talked about when we talked about endocrine functions of the GIT. It inhibits pancreatic secretions, especially the exocrine pancreas. Pancreatic polypeptide from the PP cells help in slowing down absorption of food, although the actual function of pancreatic polypeptide is not very clear. Lastly, the blood supply to the pancreas. The pancreas receives blood supply both from branches of the celiac trunk as well as branches of the inferior mesenteric, not because it has a dual embryonic origin, but largely because of its proximity with that region. So the head of the pancreas is supplied by what we call the superior pancreaticoduodenal artery. The superior pancreaticoduodenal artery is a branch of the celiac trunk eventually. It's a long route, but eventually a branch of the celiac trunk. It divides into two. We have the anterior and the posterior pancreaticoduodenal artery. Then the superior mesenteric gives you the inferior pancreaticoduodenal artery, again with the two branches, anterior and posterior. So the anterior ones communicate, the posterior ones also communicate. The communication of the superior and the inferior pancreaticoduodenal arteries is what we call the pancreaticoduodenal arcade. So that arcade supplies the head of the pancreas. Then the body and the tail of the pancreas are supplied by pancreatic branches of the splenic artery. Pancreatic branch of the splenic artery supply the body and the tail of the pancreas. That is the blood supply to the pancreas, and that marks the end of that lecture on exocrine organs of the digestive system. So our next topic will be focusing on development and malformations of the digestive system, and we'll be focusing largely on the hollow GIT first. Thank you very much. We will stop there.